Hello, everybody. We are extremely fortunate today to have a visit and the lecture you're about to hear from Professor Dr. Michelle Monje, hailing from Stanford University. Michelle graduated from Vassar, and then she, luckily for us, came to Stanford and got a MD, PhD diploma a few years ago. And then she went off to Boston and did a bunch of doctor stuff. Harvard and Mass General, I think. And then she came back to Stanford and worked with Phil Beachy. And I think actually checked the box of doing a little work on flies. Yeah, she saw the flies. She worked with a famous fly biologist, which is, everybody should have at least that little piece of Drosophila biology in their tucked away in their brains. And since then, let's see, up to Phil Beachy. Next stop is faculty at Stanford. Yeah, Stanford is also very fortunate to have her there most of the time. We've got her for today. So let's see, I'll tell you a little bit more about Michelle. She is a mother of 4. She is a prolific and successful sourdough bread baker. And she is a genius, according to John D. and Catherine T. MacArthur, and just about everybody else who's ever talked to her. And I could go on, and I'll say one thing about her scientific career and her activities over the last 10 years is an amazing body of work that I think will inspire all of us to see the real value of the work in basic neuroscience that most of us are doing, and its applicability, which is hitting the road to understanding some of the nastier human diseases. So, everybody has health and disease in their title on the grant, and everybody pays lip service to it. But Michelle, more than just about anybody else I can think about, has knit together a fascinating story with a lot to bring to both of those parties. So I'll let Michelle have as much time as possible to tell you the story herself. Thank you.
So much, and it's so wonderful to be here. As Stephen said, I'm a neuroscientist. I'm also a practicing pediatric neuro-oncologist, and so I want to tell you a story that kind of weaves together both of those kind of lines of academic inquiry. These are my disclosures. And so, broadly speaking, my laboratory works hard to understand the molecular language that cells use as they work together to build and to remodel the brain. And I want to tell you one story today in 2 parts about the way in which neurons in an activity-dependent way interact with the glial cells that form the myelin sheath. And as a field, how we're beginning to understand that this is a sort of adaptable, adaptive and plastic process that shapes and tunes neural circuits in important ways, not only during development, but in ongoing ways important for learning, memory, and other neurological functions. And then how these really powerful interactions between neurons and glial cells can be subverted, can be hijacked in the context of glial malignancies like glioblastoma and other kinds of glial cancers that occur, particularly in the childhood nervous system. But I like to start every talk with a broad consideration of neural development, and I think the best way to do that is to show you pictures of my children. So this is my daughter, Sophie, when she was an infant. And, by the time we had Sophie, who was our 4th child, we were, very aware of the amount of neurodevelopment that humans have to accomplish every day in the first year of life and beyond. And so we wanted to, help Sophie keep track of her neurodevelopmental to-dos. So we made her a checklist because we just thought, how can anybody keep up with all of the neurodevelopment that has to be accomplished every day in the human postnatal period? So, we thought the only place to put that safely was on a onesie. So we have these onesies made. But it really is pretty amazing when you consider that humans make, on average, something on the order of 500 billion new synapses every day in the first year of life. 14,000 or so new neurons in the hippocampus, 127 million new granule cells in the cerebellum, which is a structure that chiefly develops in the postnatal period. But as impressive as all of that is, perhaps the largest task that humans have in the postnatal period is to myelinate their central nervous system. We are a semi-altricial species. We don't just sort of get up and walk away the way horses do. And actually part of that is that humans are born with very little myelin in their nervous system. So myelination begins just before birth. It begins in kind of the center of the brain around the central sulcus. And then there's a massive wave of developmental myelination in the first year of life. And that myelin then progresses towards the poles of the brain. It begins in the cervical spinal cord and progresses down to the lumbosacral cord. But beyond that initial wave of myelination, there is actually a very, very long developmental process, very protracted, spanning about 30 years of human neurodevelopment. And so looking out at the audience, I think I see people here who might not even yet be done with developmental myelination. And so this long 30-year developmental process, fortunately, for us trying to study it, does follow some predictable patterns. In general, there is myelination of more basic neural circuitry, such as that underlies sensation and movement, prior to the myelination of more complex neural circuitry, such as that underlies, higher cognitive function. So, for example, there's a discrete wave of developmental myelination in the corticospinal tracts and the brainstem in the mid-childhood period, around the time that kids attain a sort of stepwise improvement in their motor milestones. And those, then later on in development, in adolescence and young adulthood, there are discrete waves of myelination in the neocortex and intercortical projections. And then beyond this developmental process, myelination actually doesn't end. The process of myelination, in which oligodendrocyte precursor cells proliferate, generate new oligodendrocytes, and ensheath the axonal membrane to decrease transverse capacitance and increase the speed of neural impulse conduction down the axon, as well as to provide metabolic support to the axon. It doesn't end at the end of this 30-year period, but actually continues throughout the lifespan in certain regions of the nervous system, and in particular in the neocortex. And actually this historical lithograph shows really clearly that there's this accumulation of myelin, and we now know from live imaging studies in rodents that there's also an accumulation of new oligodendrocytes in the neocortex over the lifespan. And so this process of myelination, I think, is just inherently really fascinating, and we should study it for its own right. But it's also really important to understand because the spatiotemporal pattern of glial malignancies in childhood, adolescence, and young adulthood actually maps really well onto the spatiotemporal map of developmental myelination, such that at a time when there is a discrete wave of myelination in the ventral brainstem around mid-childhood, between ages 6 and 8, this is exactly when one of the worst human malignancies, a cancer called diffuse intrinsic pontine glioma, tends to occur in kids of that age. And similarly, at a time when there is a discrete wave of developmental myelination in the neocortex and its projections in adolescence, this is when hemispheric high-grade gliomas tend to occur in that age group. And these observations are concordant with findings from my laboratory and from many others that these glial malignancies that we have historically thought of as quote-unquote astrocytomas because of their morphological appearance on H&E staining under a microscope, in fact, come in most cases from precursor cells in the oligodendroglial lineage, either bona fide OPCs, oligodendrocyte precursors, or just earlier precursor cells destined to go down the oligodendroglial differentiation pathway. And it certainly fits with our newer molecular understanding of these cancers that at the single cell level very closely resemble normal oligodendroglial cells molecularly. And so we may, glean really important lessons about gliomagenesis and glioma pathophysiology by better understanding what normally regulates the proliferation and functional differentiation of healthy OPCs. And so this begs a very basic question. What does regulate the proliferation and functional differentiation of oligodendroglial lineage precursors? Well, one idea that's been in the literature for some time, first introduced by Ben Barres when he was a postdoctoral fellow with Martin Raff, and then supported by some really beautiful in vitro work by Doug Fields and others, is the idea that neurons themselves may regulate the extent to which their axons are myelinated, and that they might do this in an activity and experience-dependent manner. This was a really controversial idea for many years in the myelin field, in part because there are also very clearly activity-independent modes of myelination. If you put an oligodendrocyte in a petri dish with any appropriately sized fiber, inert nanofibers, formalin-fixed axons, it'll myelinate anything of the right diameter. And so we know that there are activity-independent modes of myelination and that that contributes importantly to development. But the question remained, in the intact nervous system, could there be experience and activity-dependent modulation of that myelinated infrastructure that might be important for tuning the dynamics of a neural circuit? And so when I started my laboratory at Stanford in 2012, this was a question I was really fascinated by and wanted to answer. And fortunately, there were at the time very new tools of basic neuroscience that we could apply to begin to ask these questions. And so I want to focus the first part of this talk on the idea of myelin plasticity, of activity and experience-dependent modulation of the myelinated infrastructure of the nervous system. And to introduce you to some of the people who've led this work, this is Erin Gibson, who I was incredibly fortunate to recruit as my very, very first postdoc, now a long time ago, 12 years ago, or 13 years ago. And Erin is now an independent assistant professor at Stanford, leading her own research group. And if there's anyone in the audience here or on Zoom really interested in myelin biology, and she studies the circadian regulation of myelin, she's just an amazing, amazing person, amazing mentor, and amazing scientist. Similarly brilliant is Anna Garrity, who was a postdoctoral fellow with me, and I've been lucky enough to keep on as a senior staff scientist. So she's still working with me on questions of myelin plasticity in the lab. Juliette Knowles, who is a pediatric epileptologist, who did her postdoc with me together with John Huguenard, and now is an assistant professor at Stanford in neurology. And Belgin Yeltsin, who is a just amazing postdoc, presently on the job market. I just throw out there in case it's relevant. She is absolutely brilliant. And what Erin and Anna and Juliette and Belgin and many others in the lab have done is to leverage tools of modern neuroscience to probe these interactions between neurons and other glial cells. And so, as I'm sure this audience knows very well, this is in vivo optogenetics, which allows for control of targeted populations of cells expressing these light-activated, light-gated ion channels. And so this is channel rhodopsin 2, kind of the classical optogenetic tool, and it is responsive to blue light. It's a cation channel, and if we express it in cortical projection neurons in the mouse brain, here in the premotor or M2 cortex, and then deliver blue light at the surface of the brain, this stimulates action potentials in these cortical projection neurons. And if we do that at the frequency at which these neurons typically fire, we can begin to ask questions about how other cell types within the stimulated, oh, sorry about that, sudden activity of the mouse, but a stimulated circuit, how other cell types respond to changes in activity within the circuit. And we know that we've recruited physiomimetic activity if when we pulse the light, the mouse begins to change its behavior, in this case, execute complex motor behavior because we're stimulating the motor planning area. And for a mouse in a cage, that often looks like walking in a circle. What we found a number of years ago now is that cortical projection neuronal activity rapidly recruits the proliferation of oligodendrocyte precursor cells and just earlier pre-OPCs, again, specifically within the stimulated circuit, and even more specifically in the corticocallosal projections. We interestingly saw no effect in the corticofugal projection, so already hinting at some heterogeneity and cell type specificity to plasticity of myelin within the nervous system. If we fate map those proliferating cells using the thymidine analog, EDU, delivered at the time of optogenetic stimulation, and then look several weeks later, we find that those proliferating cells generate new oligodendrocytes. and that the myelinated ultrastructure of the circuit changes, and it changes in a way that we would predict would alter circuit dynamics and perhaps influence function. And in this set of experiments, we found that indeed motor behavioral performance improved in a way that depended upon the generation of new oligodendrocytes. Now, what are the molecular mechanisms mediating these interactions between neurons and oligodendroglial cells, at least in the premotor projections here? We thought that there might be an important role for brain-derived neurotrophic factor signaling to its receptor TrkB on oligodendrocyte precursor cells. This was a molecular hypothesis that was actually inspired by parallel work we were doing in gliomas and appreciating a role for BDNF in glioma pathophysiology, but also because, as I think this audience knows well, BDNF really does quite a lot with respect to plasticity and development in the nervous system. And so we tested this molecular hypothesis using two different genetically engineered mouse models. In the first model, made by Mike Greenberg's lab at Boston Children's Hospital, there are normal baseline levels of BDNF, but he has deleted the Krebs binding site in promoter 4 of the BDNF gene. So there's no activity-dependent increase in BDNF expression or secretion. And in a second mouse model, rather than modulating the BDNF ligand, we instead modulated the receptor for BDNF TrkB just in oligodendrocyte precursor cells in an inducible way. And so in either of these genetically engineered mouse models, either modulating the ligand or the receptor, when we repeated that optogenetic stimulation experiment, we found that in contrast to BDNF wild type or TrkB OPC specific expression of TrkB wild type mice, in which we see this expected increase in the proliferation of oligodendrocyte precursor cells in the cortico-callosal projections of the premotor circuit, that proliferative response is entirely abrogated in the absence of activity-regulated BDNF expression and secretion or OPC-specific expression of TrkB. And just as there was no proliferation of the oligodendrocyte precursor cells, there was no new generation of oligodendrocytes and no appreciable myelin changes within the circuit in response to activity. So while we think that the mechanisms mediating these adaptive changes are far more complex than just this one signaling pathway, this is a required component, at least in cortical projection neurons, and we're now finding in other regions of the nervous system for these activity-dependent changes. And so that gives us a molecular handle to begin to ask questions about what role myelin plasticity may play in various neurological and neuropsychiatric functions. And so, we and many others now in the field using this or similar strategies to block activity-dependent changes in myelin-forming cells have found that neuronal activity evoked either by optogenetics or chemogenetics or changes in experience contribute to a range of functions that include motor function, motor learning, attention, memory encoding and short-term memory, memory consolidation relevant to fear and spatial learning, as well as some forms of reward learning. There are a couple of different modes of myelin plasticity. So far, I've been talking about de novo myelination, the generation of a new oligodendrocyte and establishment of new myelin internodes on either previously unmyelinated axons or unmyelinated regions of axons. We now appreciate that particularly in the neocortex that there's discontinuous myelination and lots of opportunity to add a new internode in a way that might change circuit dynamics. In addition to this de novo myelination, there's also myelin remodeling. We know from live imaging to photon microscopy studies of the superficial cortex that myelin can change in its geometry. The internodes can become either longer or shorter, fatter or thinner, and that these even relatively small changes in the geometry of an internode can have important influences on conduction velocity and therefore circuit dynamics. As a field, an intense area of investigation right now are the molecular mechanisms that mediate these interactions. And we know that there's broadly at least two categories of mechanism mediating myelin plasticity. There's activity-regulated paracrine factor signaling, and I told you about BDNF to TrkB signaling. Charles Franz-Constance groups also found that in superficial cortex and the prefrontal cortex that endothelin is an important paracrine mechanism mediating changes in myelin remodeling. But in addition to these, paracrine signaling interactions, there are also fascinatingly bona fide synapses that form between axons and oligodendrocyte precursor cells. These were first described by Dwight Burgles in the year 2000, actually when he was a postdoctoral fellow in the JAR lab. And then, beyond that have been beautifully characterized by Dwight and by many others. There are both glutamatergic and GABAergic synapses. We've done, circuit mapping, and it really is impressive, the extent of connectivity of OPCs. They are listening to the circuits that they are part of. But the role of these well-characterized neuron to OPC synapses in myelin plasticity is still not well established. We really actually don't know the function of the synaptic input into the OPC, and that's a major open question in the field. What is clear, both through computational as well as experimental modeling, is that in the healthy nervous system, plasticity of myelin generally promotes coordinated circuit function, such as synchronous oscillations between nodes in a network. And so if myelin plasticity is, in the healthy nervous system, adaptive and responsive to experience and contributes to coordinated circuit function, important for, a range of functions like attention, memory, and learning, it stands to reason that when myelin homeostasis and plasticity is impaired, that this may contribute in really important ways to diseases of cognition. And as a pediatric neuro-oncologist, the disease that my laboratory has studied for many, many years is cancer therapy-related cognitive impairment. So I'm going to tell you a little vignette about how impaired myelin homeostasis and plasticity contributes to that chemotherapy, so-called brain fog. I'll tell you that there are some similar and parallel mechanisms at play in the cognitive impairment syndrome that we're presently grappling with after COVID, so-called COVID brain fog. So let me tell you those vignettes. After cancer chemotherapy, there's a really consistent and debilitating, persistent syndrome of impaired attention, memory, speed of information processing, and multitasking. This affects a large fraction of patients after a range of different chemotherapeutic agents, but some are more prone to cause this syndrome than others. And one particularly bad actor is an antimetabolite called methotrexate. So we developed a methotrexate mouse model to try to understand the underlying neurobiology of this kind of enigmatic syndrome. And what we found was that systemic methotrexate chemotherapy administered to mimic high-dose regimens of methotrexate directly activates another kind of glial cell called microglia. And that, interestingly, this isn't a pan-microglial response. It's a specific subpopulation of microglia just in axon tracts, just in the subcortical and hippocampal white matter. These reactive microglia, in turn, activate astrocytes through cytokine signaling mechanisms beautifully described by Ben Barres and Shane Lidlow. And together, these reactive microglia and astrocytes dysregulate the oligodendroglial lineage and impair both myelin homeostasis and entirely block myelin plasticity. Activity-regulated myelin response is gone. And all of this dysregulation is persistent. In the mouse model, we looked up to 6 months later. In humans, we know that this cognitive impairment can persist for years to decades. What we discovered was that the reactive microglia were really central to this pathophysiology. If we deplete microglia transiently, this normalizes the state of astrocyte reactivity, and it restores the oligodendroglial lineage and myelination. It also rescues the cognitive impairment that we find in these mice. And so understanding that, in the context of chemotherapy-induced brain fog, microglia are a really important part of the pathophysiological picture. I was really worried when the pandemic struck because it was clear how profoundly immunogenic this virus was. And I was worried we were going to begin to see a syndrome that looked a lot like chemotherapy-induced cognitive impairment. And very early, by the summer of 2020, people were really flooding neurologist offices complaining of precisely the same symptoms: impaired attention, memory, speed of information processing, and multitasking. And so I reached out during the depths of the COVID shutdown, when the lab was not even functioning, to Akiko Iwasaki, who who is this really wonderful thought leader in COVID virology and immunology, as well as Abhinath at NIH, who studies sort of neuroviral syndromes. And together, we began a collaboration that once the lab reopened, we were able to really kick off using a mouse model that the Iwasaki lab had developed, in which there is respiratory restricted infection by SARS-CoV-2. We wanted to test the hypothesis that it is the inflammatory response, the immune response to the infection in the respiratory system, not direct brain infection, that is contributing to this brain fog syndrome, or at least one of the ways that COVID can influence the nervous system. Certainly, there are many. And so in this model system, there is no brain infection. There is no infection outside of the respiratory system. Mice actually don't get very sick. They groom well, they run around the cage, they don't lose any weight. They clear the virus from the respiratory system within a week. And yet, when we look even 7 weeks later, we see the exact same pattern of axon tract microglial reactivity. And as we would predict, there's a associated dropout of oligodendrocytes, about a 30% decrease in the number of oligodendrocytes in subcortical and hippocampal white matter, and a decrease in the number of myelinated axons. This happens very quickly. We see it by 1 week after infection, and it persists through 7 weeks after infection. When we compare the degree of myelinated axon dropout after exposure to systemic chemotherapy or after this mild respiratory COVID model exposure, it's pretty similar. So kind of fitting with the similarity in the clinical syndrome. Okay, so we think that myelin plasticity contributes in important ways to attention, memory, learning, and other domains of cognition. That loss of myelin homeostasis and plasticity is important in diseases of impaired cognition. But what happens if there's too much adaptive myelination? What happens in diseases that are characterized by abnormal patterns or abnormal amounts of activity? Might myelin plasticity actually contribute to disease when it is upregulated in those contexts? And so we have studied this now in the context of a generalized epilepsy syndrome, as well as in reward learning in the context of drugs of abuse. And just in the interest of time and focus, I'm just going to tell you about one of these vignettes, but would be really excited to tell you in later meetings or in the Q&A section about the more recent work in reward learning. So in work led by Juliette Knowles and in collaboration with John Huguenard, we wondered whether myelination could be maladaptive in the context of generalized epilepsy, whether adding too much myelin within a seizure network might promote the hypersynchrony that characterizes these generalized epilepsy syndromes. And indeed, when we looked at 2 different rodent models, both a rat model and a mouse model that I'm showing you here of absence epilepsy, we found that there was exactly this increase in myelination that was specific to the seizure network and evident only after the onset of seizure. So this is a seizure syndrome that affects thalamocortical circuitry. And within those thalamocortical networks, but not outside of them, we see after the onset of seizures, this increase in myelinated axon number, as well as in the thickness of the myelin sheath. This depends upon the seizures. If we treat the mice with ethosuximide, which in this case prevents the seizures from beginning in the first place, then we do not see these myelin changes in these genetic mouse models of epilepsy. And so then the question was, does this aberrantly increased myelin promote increased synchrony between nodes in the network and exacerbate the seizure syndrome, which is what happens in the natural history of the disease? The seizures become more frequent with time and more severe with time in untreated animals as well as in patients. And so we tested this by blocking activity-dependent myelination, in this case, utilizing the OPC-specific inducible TrkB knockout mouse model. And we found that in the absence of the addition of new oligodendrocytes in response to activity, that there was a decrease in what's called ictal coherence, which is the synchrony between nodes in a neural network during the seizure. And as we had hypothesized, this prevented the worsening of the seizure syndrome over time. In mice with intact myelin plasticity, the number of seizures that occur over the sort of course of the disease increases with time, and that exacerbation is really markedly blunted in mice that lack the ability to respond to neuronal activity by making new oligodendrocytes. So we think that myeloplasticity is important and that it contributes to disease both when there is not enough of it and when there's too much of it. But what happens in the context of brain cancers? What would happen if that OPC had an oncogenic mutation? Could these really powerful proliferative signals between neurons and normal OPCs be hijacked and subverted in the context of glial cancers? So I'd like to focus the second half of the talk on the idea of malignant myelin plasticity, of activity-dependent glioma growth and progression. And to introduce you to the people who've led this work, I do have men in my lab. I just happen to be showing you the work of some really amazing women, but it's a diverse lab. So I wanted to, just highlight the people who've really led the work that I'm about to show you. This is Hamsa Venkatesh. She was an extraordinarily brilliant cancer biology PhD student who came to my lab, and I was lucky enough to keep as a postdoc. She's now leading her own independent group at Dana-Farber Cancer Institute and Brigham and Women's Hospital. If there's anybody in the audience here or on Zoom interested in, cancer neuroscience postdoc, Hamsa's amazing, as is equally brilliant, equally amazing, equally open for postdoctoral applications, Ewan Pan, who did her postdoc with me. And after a first postdoc with David Gutman, we kind of co-mentored her, and she's now leading a group at MD Anderson as an assistant professor. And then Catherine Taylor and Tara Barron, who are both presently postdocs in the lab. Katie is on the job market this year and is wonderful. And as a pediatric neuro-oncologist, this is the disease that I and my laboratory spend a lot of time thinking about. These are gliomas of childhood, which, as I mentioned earlier, arise in a really predictable spatiotemporal pattern. In fact, if you tell me that a child has a glioma and give me their age, I will probably be able to predict where their tumor is. I want to introduce the cancers that we're going to talk about in the coming slides. This is low-grade glial malignancy called optic pathway glioma. This tends to occur in association with a tumor predisposition syndrome called neurofibromatosis type 1. It also can happen sporadically. This is a low-grade glioma. It tends not to be lethal, but is quite debilitating, and children lose vision in one or both eyes. This is a disease that tends to happen in early childhood, and I'm showing you an image here from a 4-year-old. As I mentioned earlier, this is one of the worst imaginable human cancers. This is the most common high-grade glioma of childhood, one of the most common diseases that we see in pediatric oncology clinic. It is universally lethal. It's a cancer of the brainstem called diffuse intrinsic pontine glioma. It peaks in incidence. It can happen at any age, but it peaks in incidences around age 6 and typically results in death in less than a year. A molecularly related disease that happens in slightly older children occurs in the thalamus, or often both thalami. And then this is a hemispheric high-grade glioma, also called pediatric glioblastoma. This looks radiographically very much like its adult counterpart in adult glioblastoma. There's some differences molecularly between pediatric and adult hemispheric high-grade gliomas, but they behave similarly. And all of these diseases, although I'm showing you images in which they're sort of relatively localized on the imaging to one place, they all spread throughout the nervous system in a very diffuse manner. And I think that the spatiotemporal pattern with which they emerge really underscores how much of a disease of neurodevelopment these cancers are. And the idea that neurons might have something to do with it is something actually that a very prescient and brilliant neuropathologist recognized almost 100 years ago. H.J. Shear described a very long time ago what he called the secondary structures of Shear, and which we now call perineuronal satellotosis, the propensity of these malignant glioma cells to cluster in very tight microanatomical association with neurons, both clustering in this kind of like a halo around the neuronal soma, kind of evoking images I know Joanne has been studying of these, OPCs that tend to sort of form satellites around neurons, really recapitulating that behavior, and then to invade along axon tracks, invade along white matter tracks. And this concept that, there might be something going on in this interaction, this tight, anatomical interaction that sort of hints at a really important signaling interaction, was something I thought a lot about as a trainee. I remember as a pediatric neuro-oncology fellow and as a neurology resident, just kind of staring at H&E images like this and thinking, what is going on with the neurons? Why are these tumor cells so close to them all the time? And this particular H&E image comes from a patient of mine when I was a pediatric neuro-oncology fellow, a 5-year-old boy with diffuse intrinsic pontine glioma. And it was actually this child's early postmortem tumor donation. I'm showing you an image from that enabled us to make the very first patient-derived cell culture and xenograft mouse model of DIPG. And it's really through those kinds of incredibly giving, very selfless donations of tissue, often at the time of death from our patients and their families, that enables us to do all of the work that we do. So I just want to, acknowledge that incredibly important contribution of patients and families. And so to ask the question, do neurons in a activity dependent way promote the proliferation and growth also of glial malignancies? We did that same optogenetic experiment that I have mentioned before, optogenetically stimulating cortical projection neuronal activity, but this time in the context of a diffusely infiltrating patient-derived cortical glioblastoma isolated from an adolescent patient of mine. And what we found was that when we stimulated cortical projection neuronal activity in this context, that just like their normal counterparts, the malignant glioma cells increase their rate of proliferation, and that this results in an increase in the tumor burden, again, specifically within the stimulated circuit. So brain activity can, in fact, drive brain cancer growth. More recently, and in collaboration with David Gutman's lab, we wondered whether the same was true of low-grade glioma types. And David's lab had generated a really genetically accurate and faithful model of NF1-associated optic pathway glioma, in which the tumors always emerge at 9 weeks of age. So we know exactly where and when these tumors are going to form. And if we optogenetically stimulate the optic nerve just prior to tumor initiation, beginning at 6 weeks, and then examine the tumors that form later on at 16 weeks, what we find is that the tumors from the optogenetically stimulated animals are much larger, shown here in black dots, than those in littermate controls that were identically manipulated, but in whom we just did not turn on the light. Now, this optic pathway glioma model was an exciting opportunity to ask questions not just about tumor proliferation and growth, but also about tumor initiation, because we know exactly where and when these tumors are going to form. And we actually don't need to optogenetically stimulate, this is the optic nerve. We can modulate activity in the optic nerve just by changing visual experience. And so we tried to get the mice to wear these sunglasses, and it was a disaster. There was plastic everywhere. They just chewed them up. I don't recommend that as a strategy. I'm kidding. What we did instead is we just put them into complete darkness. And so if we dark rear these animals for 24 hours a day, and we do that at the time that tumors tend to initiate, very consistently tend to initiate at 9 weeks of age, or even just after, beginning dark rearing at 12 weeks of age, when we examine the optic nerves at 16 weeks, what we find, shown here in the blue data points, is that far fewer and much smaller tumors have formed compared to littermate controls that were simply raised with normal visual experience. We wondered whether this was a circadian effect, so we did some light entrainment experiments, and that didn't change the results. This really was about the decrease in activity in the optic nerve. If instead we put the mice in darkness at 6 weeks of age prior to tumor onset, absolutely no tumors formed in the dark-reared animals compared to 100% tumor initiation in light-reared animals or normal visual experience animals. And this was true even if we put them back into regular light cycles after what appears to be a susceptibility period for tumorigenesis, and despite the very strong oncogenic predisposition that these mice have for optic pathway glioma. So there's some really powerful interactions going on. And we wanted to understand what the mechanisms were. And we first hypothesized that there were important paracrine interactions, that there may be activity-regulated growth factors, for example, that are secreted and that contribute to glioma growth. And so we did a simple set of experiments in which we took explants of either normal mouse cortex or normal mouse optic nerve and retina, and then collected the secreted factors in conditioned medium from those explants when the neurons in the explants were at varying levels of neuronal activity. So either spontaneously active, silenced with tetrodotoxin, or optogenetically stimulated. And what we found is that when we then took that conditioned medium and placed it onto cultures of different glioma cells, that there was an activity dose-dependent increase in glioma cell proliferation that was lost if the slices were silenced with tetrodotoxin. So gliomas are, as I mentioned before, sort of a family of clinically and molecularly distinct disease entities. They're not all the same disease, but we found that this response to activity-regulated secreted factors was really conserved across every different kind of glioma culture that we looked at, from pediatric, diffuse intrinsic pontine glioma to IDH wild-type adult glioblastoma and low-grade NF1-associated optic pathway glioma. So what's in the conditioned media? That's summarizing, 2 years of a lot of work and proteomics in just a single text box, as we do. We found that there were using this particular experimental setup, we discovered 2 important activity-regulated glioma growth factors. Not surprisingly, just as BDNF promotes the proliferation of healthy OPCs, it also promotes the proliferation of glioma cells in vitro. And I'll tell you, does much more important and complex things in vivo. But really unexpectedly, we found a shed form of the synaptic adhesion molecule Neuroligin-3, which we did not expect to be a glioma mitogen, but it is. So, Neuroligin-3, as this audience knows very well, is a very important postsynaptic adhesion molecule present at both glutamatergic and GABAergic synapses, first discovered by Tom Sudoff, in recent years gaining some notoriety because there's a point mutant form that contributes to autism spectrum behaviors. But it was not known to be a mitogen in any context and actually wasn't known to even be shed. But what we find is that Neuroligin-3 in the healthy nervous system is shed through cleavage at the membrane and release of this large N-terminal ectodomain. This occurs in a strictly activity-dependent manner. And again, summarizing a great deal of work in the interest of time, we found that it is activity-regulated function of the metalloprotease ADAM10 that mediates the cleavage and release of Neuroligin-3. And so the next question was, what cell types are shedding Neuroligin-3 and why? And so we did, first a look at the gene expression, of different cell types in the nervous system, and found that not only neurons, but actually also oligodendrocyte precursor cells express very high levels of Neuroligin-3. And when we conditionally delete Neuroligin-3, either from oligodendrocyte precursor cells or from different subtypes of neurons, we find it's actually OPCs that are the chief source of shed Neuroligin-3. This is where the Neuroligin-3 is being shed. And in conversations later, and in the interest of time, I won't tell you the story right now, but I would love to tell you later, we now understand that Neuroligin-3 is playing a very important role in normal myelin biology. A topic of a different talk. But, we want to understand it here in the context of glioma. And one question we had was, does it matter? There are lots of different mechanisms that gliomas have evolved to grow. There are cell intrinsic mechanisms of proliferation. There are also numerous microenvironmental interactions. So we wanted to understand the relative importance of this particular, kind of surprising mechanism of tumor growth. And to do that, we simply xenografted brain cancer cells into the environment of either the Neuroligin-3 wild type or Neuroligin-3 knockout brain. And what we found was really unexpected. Rather than even just slowing in their growth, we found that in the absence of neuroligin 3 from the brain, that there was just an utter stagnation of the tumors. Here you see these GFP labeled patient-derived glioblastoma cells. We put them into the mouse brain in a wild type mice. They grow, they expand, they invade across the corpus callosum. In the absence of neuroligin 3, even 6 months later, we just see the cells that we xenografted. They've persisted.
Is that deleted everywhere or just in the PCs?
This is deleted everywhere in the brain. And this is a knockout mouse. This mouse has no neuroligin 3. They're actually, as you know, the Sudoff lab and others have published, they're fairly behaviorally normal mice because in neurons, neuroligin 1 seems to compensate for much of the function of neuroligin 3. Question.
Sorry, related question. So
Yeah.
xenograft cells have neuroligin 3?
Yes, that's right. They do. They do. And I'll give you some unpublished data on that. Yes, they do. It turns out that there is a feed-forward effect, which I'll get to in a second, of neuroligin 3 exposure on glioma neuroligin 3 expression. So the gene is regulated by exposure in the brain. In culture, the tumor cells in early passage cultures, in vitro, there's very little glioma neuroligin 3 expression. When they are exposed to it, either experimentally or when we put them back into the brain, they then start to express into the wild type brain. They start to express neuroligin 3, but don't in the knockout brain. And we can monitor this growth, using in vivo bioluminescent imaging, so we can see it in real time. And there's just this stagnation of tumors in the neuroligin 3 knockout brain. And that's true across multiple different kinds of gliomas and in multiple different brain regions, both in the cortex and in the brainstem. What we don't see, though, is this apparent dependency on neuroligin 3 doesn't extend to a patient-derived model of breast cancer brain metastasis, suggesting that while neuroligin 3 is a really important glioma factor, it might not be important for all kinds of cancers being exposed, to the brain. So we then turned, back to this really useful and important model of optic pathway glioma. And we wondered if in the NF1 associated optic pathway glioma context, neuroligin 3 might also be an important mechanism. And we wondered that not only because of what we had learned about high-grade gliomas, but also because when we look at the NF1 mutant optic nerve, now remember, this is a tumor predisposition syndrome in which all of the cells in the body have lost one functional copy of NF1. We see that in the NF1 mutant optic nerve, there's aberrantly increased shed neuroligin 3 present. There's aberrantly increased shedding of neuroligin 3, and it's being shed from OPCs in the optic nerve. We wondered whether that was actually part of the predisposition to tumors in this particular disease context. And so we tested the role of neuroligin 3 in the NF1 optic pathway glioma model simply by crossing the neuroligin 3 knockout mouse to this optic pathway glioma mouse model. And what we found is that this largely phenocopied the effect of dark rearing, that even with normal visual experience, far fewer and much smaller tumors formed in the absence of neuroligin 3. So what is going on? Is this shed synaptic protein influencing gliomas? Well, we found that when neuroligin 3 is shed in an activity-dependent manner from the postsynaptic membrane of chiefly of OPCs by the enzymatic activity of ADAM10, it then binds to the glioma cell, not through Neurexin, but actually through another binding partner that we've just identified and happy to talk about afterwards. Again, sort of the topic of an entire talk. And once it binds, it recruits in the glioma cell numerous oncogenic signaling pathways with early upstream focal adhesion kinase, downstream SRC, RAS, and PI3 kinase pathways activated. So that helps us understand the role of neuroligin 3 as a mitogen in this context. It helps us understand the sufficiency of neuroligin 3 to promote growth, but it doesn't explain that really surprising dependency. And so we dug deeper and we looked at the gene expression changes attributable to neuroligin 3 binding in the glioma cell. And we found that there was a really strong effect on synaptic gene expression. And in fact, when we look at all the different gene expression changes that are evoked by neuroligin 3 binding in glioma cells, we see a number of different sort of classes of genes being regulated that we now understand to be very important in neuron-glioma interactions. But the first one that caught our attention were these synapse-associated genes. So neuroligin-3 binding to the glioma cell causes a strong upregulation of neuroligin-3 expression in the glioma itself, together with upregulation of the BDNF receptor, TrkB, encoded by mTrk2, and a number of other glutamate receptor and other kind of synapse-associated structural proteins. So that was interesting, and really caught our attention and made us wonder if, like there are synapses between neurons and healthy OPCs, there might also be synapses between neurons and glioma cells, and whether maybe that's why neuroligin-3 was so important, is that it perhaps was encouraging those synapses to form. When we look by immunoelectron microscopy, these are GFP-labeled glioma cells. We've xenografted into the mouse brain so we can do immuno-EM for the GFP and thereby unambiguously identify the tumor cells. We see these really clear synaptic structures with presynaptic neurons containing synaptic vesicles, a synaptic cleft, postsynaptic membrane on the glioma cell. And to test the role of exposure to neuroligin-3 in mediating these, we see far fewer of these structural synapses form in the absence of neuroligin-3 from the tumor microenvironment. But are these structural synapses between neurons and glioma cells just a shadow of the cell type from which we believe these tumors emerge, or might they actually be electrophysiologically functional? So to test that, we collaborated with Rob Malenka and did whole cell patch clamp electrophysiology of tumor cells xenografted to the hippocampus, just as a very, experimentally tractable circuit. We put these GFP-labeled tumor cells in CA1 region, and then we make acute, 8 weeks later, after the tumors have been grafted, we make acute hippocampal slices, and then we can record from the glioma cells, and at the end of the recording, fill them with biocytin, go back and just confirm that we were indeed recording from a glioma cell and not accidentally patching onto a normal cell. And what we find when we do this experiment is that in a subset of glioma cells in each patient-derived tumor we examined, there are really clear EPSCs, really clear excitatory postsynaptic currents. These depend upon action potentials. They're blocked by tetrodotoxin. They exhibit multiple electrophysiological characteristics of true synapses, including paired pulse facilitation and in the presence of strontium, mini EPSCs. And this first kind of synapse that we found were mediated by calcium-permeable AMPA receptors. Now, I have to tell you, this seemed crazy to us. It really, I had a pause and said, could this be real? And I have to say this was one of the most wonderful, times when our work was sort of immediately replicated. I found out that a colleague of ours in Germany had discovered the exact same thing. We co-published the papers together. We independently found this, and that made me feel a lot better about the whole thing. As we know, synapses in the healthy nervous system, particularly AMPA-mediated synapses, exhibit mechanisms of adaptive plasticity. And one of the main ways that happens is mediated through NMDA dependent mechanisms. It turns out that the glioma cells don't express NMDA receptors, but they do express high levels of TrkB, of mTrk2. And so we wondered whether another mechanism of adaptive plasticity at synapses, a less well-known one mediated by BDNF to TrkB signaling, might be at play in the glioma cells. And indeed, when we patch clamp, the glioma cells and record from them in the presence of BDNF, the amplitude of the postsynaptic current increases. And this depends upon BDNF receptor TrkB expression in the glioma cell. If we CRISPR delete mTrk2, which encodes TrkB, then we don't see this effect of BDNF on the magnitude or the amplitude of this current. And just in the interest of time, to summarize a very recent story that has come out, what we have found is that the glioma cells are really hijacking this classical mechanism of postsynaptic plasticity, and through TrkB to CAMK2 signaling, increasing AMPA receptor trafficking to the postsynaptic membrane, thereby increasing the amplitude of the current. Now, there are other neurotransmitter receptors expressed in glioma cells, and we wondered whether there might be GABAergic synapses as well. While the AMPA-dependent synapses are expressed in adult GBM, as well as in pediatric versions like diffuse intrinsic pontine glioma and pediatric GBM, we see different expression patterns of GABA. There seems to be less uniform expression across different tumor types, where there's high expression in DIPG, this characterized by this particular mutation, H3K27M, as well as in an IDH mutant molecular subtype of adult GBM, but not in sort of the more classical IDH wild type glioblastoma. When we examine diffuse intrinsic pontine glioma cells or IDH mutant glioblastoma cells, we see really clear GABAergic currents. These are mediated by GABA A receptors. They're blocked by picrotoxin as well as by bicuculline. And they're augmented by allosteric modulators of GABA receptors like benzodiazepines, like lorazepam. Now, we think about GABA as an inhibitory, hyperpolarizing neurotransmitter, but in immature neurons, in oligodendrocyte precursor cells, GABA is instead depolarizing, and of course, it's just a chloride channel. So, it all depends upon the intracellular concentration of chloride in the tumor cells. And we find that GABA currents are depolarizing in the tumor cells, and again, very specifically in diffuse intrinsic pontine glioma, but not in IDH wild type glioblastoma. And this is due to the high intracellular chloride concentration within the tumor cells, and that in turn is mediated by expression of the chloride transporter NKCC1. We also have some structural evidence of GABAergic synapses in, this is hot off the press data that I'm showing for the first time, between neurons and glioma cells in co-culture. So if we co-culture neurons together with patient-derived pediatric glioma cells, and we express in those glioma cells a GFP-tagged version of the GABA subunit GABA-G2, then we see synaptic puncta co-localization between neurofilament marked axons and presynaptic synapsin, and then glioma process marked by nestin and this GFP tag. There's I just like this movie because we just found it. And so that would suggest then that GABAergic interneurons might be promoting the growth of these pediatric glioma types. And indeed, if we optogenetically stimulate GABAergic interneurons in the mouse brain, then that increases the rate of proliferation and growth of the pediatric glioma cells. Which made me worry because we give benzodiazepines like lorazepam to these kids for various reasons. And what we find is that in vivo, if we treat mice bearing these pediatric tumors with lorazepam, that there's an increase in tumor cell proliferation and a decrease in mouse survival. And so, while we need to identify the drugs that interrupt neuron-glioma interactions and perhaps leverage them for therapy, we also have to be careful about drugs that might conversely be damaging in this context. So in summary, and I have 5 minutes left. I'm going to finish, I promise. These cancers are integrating into the neural circuits that they invade. They do this both through bona fide neuron to glioma synapses that are then amplified and elaborated through mechanisms of adaptive synaptic plasticity. They're also activity-dependent potassium-evoked currents that are in turn amplified through tumor-to-tumor gap junctional coupling. And that's a lot of different ways that tumor cells can undergo membrane depolarization, which is a very metabolically expensive thing to do. And so it suggests that membrane depolarization itself might be important, evoking some voltage-sensitive mechanisms of cell proliferation. And we tested that idea, again, using optogenetics, but this time expressing the light-sensitive opsins not in the neurons, but rather in the tumor cell. And so then we can use blue light to depolarize the tumor cell. And if we do that in vivo, we see that it increases the rate of tumor cell proliferation. But the tumors aren't just undergoing membrane depolarization. They're also, incorporating mechanisms to increase the magnitude of that depolarization. And we wondered whether that also mattered. So using different optogenetic strategies to either cause a medium-sized membrane depolarization or a larger membrane depolarization, we see that the larger membrane depolarization promotes the proliferation. Conversely, if we block these interactions, in this case by expressing a dominant negative AMPA receptor subunit, but also using very specific AMPA receptor targeting pharmacological strategies, that has a very stark growth inhibitory effect. We can visualize this electrical activity within the tumor cells using genetically encoded calcium indicators. And I think this particular calcium imaging movie of a diffuse intrinsic pontine glioma really underscores this new understanding that these cancers are electrically active tissues. And that's not how we have been approaching them either in terms of understanding them or treating them. And so now we're working hard to understand the mechanisms of malignant circuit assembly, plasticity, and evolution over the disease course, to discern the granular details of these voltage-sensitive mechanisms of tumor cell proliferation. And we suggest that in doing so, we may glean important insights for normal neuroplasticity and development kind of viewed through this magnifying lens of glial cancers. To end kind of with a summary, from our work, but now also from many others in the field, we find that there is this cycle of interactions between neurons and glioma cells by which, neuronal activity of multiple different neuronal types drives glioma cell proliferation, likely resistance to treatment and invasion of the tumor, both through paracrine as well as synaptic mechanisms, where in turn, the glioma cell remodels the neural circuits to better feed the tumor and also causes hyperexcitability that contributes to really driving this malignant cycle. And as we understand these mechanisms, there are a number of different potential therapeutic targets that emerge and some clinical trials beginning to target these neuron-glioma interaction targets. The mechanistic parallels that are really evident between normal neuron-glial and malignant neuron-glioma interactions underscores the extent to which these cancers are just hijacking mechanisms of normal development and plasticity and really demand that we approach them from a neuroscience perspective. Lots of people to thank, lab members, past and present, collaborators, funding sources, and last but not least, the patients and families whose donation of tumor tissue really enables all the work that we do. Thank you so much for your attention, and hopefully we have a minute or two left for questions.
Well, I've got about an hour's worth of questions. So, but first, who else has a question? So.
Thanks. This is really wonderful work and very sweeping and yeah. So these parallels to synaptogenesis, as you probably know, right? Neuroligin expressed in any cell type generally can cause sort of ectopic generation of synapses. This sort of part of the story here. And do you think we learn actually something about normal synaptogenesis, this paracrine release of neuroligin, which induces expression of neuroligin, could be sort of something that, I don't know if neurobiologists are following up on this possible mechanism for early synaptogenesis in neural circuits. Again, there are many other questions, but this is one I have. Yes.
Yeah, absolutely. I think it's a really important point, and I think it highlights how sometimes the cancer biology can lead us in important physiological, questions. I think one of the things that I've been really surprised by is how much of the neuroligin 3 is actually coming from oligodendrocyte precursor cells. There's some being shed from neurons, but a major shedding source are the OPCs. And what we're finding is that, it's a really cool story that, ends up involving some very interesting signaling and piezo, and it's a topic of another talk, but we find that that's a really important mechanism to maintain the stemness of the OPC and maintain an appropriate population for to be available for these plasticity mechanisms. And so I think the question in my mind is whether, or there are many questions in my mind, but one is whether that shedding also is important in modulating release of those, enigmatic neuron to OPC synapses to allow that OPC to then proliferate or differentiate. And the role that the shed neuroligin 3 is playing in at normal neuronal synapses is an open set of questions.
Thanks. This is a really interesting talk. I was curious, you went over it a little fast, so the slide where you're doing the experiment, activating GABAergic neurons,
Uh-huh.
that's also a technique people use to shut down neural activity on the excitatory cells. So there's both an increase in GABA release, but then presumably a decrease in the glutamatergic
Yeah.
synapses that you were.
Yes,
So I'm
that's
curious
true.
which of those I don't know, which of those factors you think is more driving the increase in proliferation?
We have found there, so in the tumor, about 10% of the tumor cells in a given tumor have the glutamatergic synapses, at least as we can determine, but about 30 to 40% have the GABAergic synapses. So I think in that particular tumor type, at that stage of disease, the GABAergic signal is actually more important. Yeah, but it's a great point. And we also, to follow that up, when we look at the effect of broad acting anti-seizure medicines, we don't see much of a therapeutic effect, but when we use those that specifically disrupt either the AMPA receptor dependent signaling, or we've actually found one that disrupts in a tumor specific way, the GABAergic signaling, that has a really, really large effect on tumor growth. It really slows it down. So I think targeting those specific interactions is more important than overall decreasing excitability and generalized neuronal activity.
Hi, thanks for the great talk. Thinking about your data where you were showing the effect of light or activity on the growth of these tumors, I'm wondering, would you expect to see anything different in animals that are primarily active, like during the daylight, or more diurnal animals?
That's a great question. So we think that, there's activity during sleep, there's activity, during wakefulness. We don't yet know how different patterns of activity differentially influence the glioma. But there's also really important circadian regulation of these cancers. And that's a really open and early area of research. Very important. There's very important glioma circadian biology to be better understood.
That was a wonderful talk. You mentioned that there was a difference in activity-dependent myelination depending on what white matter tract you were in.
Yeah.
I was wondering if you could comment a little bit more about, you made a comment that maybe there's some diversity in OPCs and oligodendrocytes.
Yeah, so
expand on that.
This is just a wide open, new and important concept, I think, in the field. So there seem to be different principles in different circuits and real specificity as not just to which neurons get myelinated, but which regions of the axon get myelinated. So I get very excited thinking about this. We really have so much to learn. But for example, decreasing sensory input to to the visual cortex, causes, Paula Arlada has shown, not so much changes in cell number, but much more in the morphology of the internodes. So, really active remodeling of the internodes in response, and only on GABAergic axons and not on glutamatergic axons. And, different kinds of sensory deprivation, whisker trimming, for example, in looking at somatosensory cortex, has a different effect and really in that part of the cortex seems to influence oligodendrocyte number, Ethan Hughes's work. So there seem to be different rules in different places. We found in a couple of different circuits that, corticocallosal axons, evoke this activity-dependent myelination. Corticofugal ones do not. In the dopaminergic reward circuitry, dopaminergic axons get myelinated in an activity-dependent way, but only in the proximal segment. So, and so there's just some really interesting rules that seem to be circuit-specific and need to be uncovered experimentally.
One more question.
Sure.
Fine for me.
The first question. In your Neuron-glial 3 models, do you ever look at two things? The first is any changes in myelination. Second is any synapses formation. You were mentioned earlier that there's unknown cause of we don't know what's the cause of the oligodendrocyte that's form synapses to the neurons and the other way around. Do you ever see any changes of those similar to blastema formation?
So we're looking right now at some OPC specific Neurolink 3 knockout models. And there does seem to be a decrease in the number of oligodendrocyte precursor cells, particularly earlier in development. And what we're finding, just to quickly sort of mention this story, is that Neurolink 3, when it is shed, binds to CSPG4 and G2. and that causes a change in membrane tension that opens up piezo-1. And the piezo-1 mediated depolarization triggers something internally, some signaling pathway we don't yet understand, that maintains the cell in a non-differentiated state and promotes stemness. And so we're learning about the role that this interaction has in, the decision to differentiate or to maintain stemness. How that plays out, I think there are lots of redundant roles, to make sure that the nervous system gets myelinated. We don't see that there's a broad lack of myelination in Neuroligin 3 knockout mice, but we're right now examining their activity dependent responses and ability to remyelinate in different contexts. So lots of work still left to do to understand it.
Okay, well, let's see. Nuno was here only virtually today because he's got a cold. He's staying home with a cold, but he did have a couple questions and
Okay.
they're related. And one was about the shed neuroligin 3
Mhm.
signaling, and the other one was about the BDNF signaling. And he just wondered if you had any ideas about how those were localized on subcellular compartments. Is it somas, distal processes?
Very, very good question, and something that I would love to, work together to better understand. We don't know. There's some evidence that the neuron to glioma synapses are happening at long glioma processes, so that act a little bit like dendrites, but we don't know that doesn't exclude that there are somatic synapses as well. We've chiefly seen them in processes. As for within the OPC localization of Neuroligin 3 signaling, it's really happening in an autocrine and paracrine fashion with CSPG4, which is kind of everywhere. But I don't know, because we haven't done any dynamic imaging, whether that's really happening on the OPC processes or on the soma. And the BDNF signaling, we also don't know where it localizes. When we do, TrkB, we see puncta throughout, both the processes and the soma, but I don't know where the important signaling is occurring. So lots that we don't know.
Yeah, well, do you or Tom Sudoff or anybody think does the shedding only happen? Do you have to have a synapse? Neuroligin 3 have a synapse to get shed, or could it be shed in a way that doesn't have that much to do with synapses?
It can be, I can say that it can happen in a way that doesn't have much to do with synapses, because we see shedding that can be evoked with OPCs in culture.
Which don't have synapses.
Which don't have synapses because there's no neurons. There are no neurons around. Yeah.
Okay, well, I have been saving up a couple burning questions myself. And one is, it's really on the borderline of the two halves,
Okay.
two parts of your talk.
Yeah.
And let's see, you left a big question. You talked a lot about BDNF being part of the activity dependence of myelination and OPC proliferation in normal development and so on.
Yeah.
There are synapses there, but you had a big question mark on your slide about the functional importance.
Yeah.
And so questions remain, or you would have said a little bit more. But what I'm wondering about at the borderline between the two is is it possible that those synapses have to do with the transition from the healthy state to the disease state? In other words, to the initial ignition of the glioma.
Yeah. Very possibly. It is very possibly the case. We don't know. We just don't know.
And then if they do, does that mean that people should stay away from benzodiazepines?
No, in general, no. If you are a patient with an H3K27M mutant diffuse midline glioma or an IDH mutant hemispheric high-grade glioma, benzodiazepines have a role, but should be thoughtfully and sparingly used, I think.
And the Z drug, zolpidem, does that do the same thing? Is there any data on that?
So I don't know about the GABA B, the role of GABA B.
No, this is presumably acts on one of its actions is the same, it's a benzodiazepine receptor, but it's a chemically different compound.
Yeah.
So it might tease apart, whether it is actually the GABA A enhancement role of the benzodiazepine or something else.
Using a lot of different tools to modulate GABA A receptors, I do strongly think that that's one very important component of the pathophysiology in that tumor type, but not in others.
And then since not everybody has left yet, what about, I'm sorry. I thought go ahead.
Very interesting talk. I was wondering if you know whether the shed neuroligin 3 has any effect on the neurons, and more generally, if OPCs are talking back to neurons in some way.
It's a great question. I think, so number one, I have no idea what the shed neuroligin 3 is doing in neurons, because I think there are multiple potential binding partners. The chief one that we find in glioma and in OPCs is unpublished, but CSPG4, which is NG2, which is very strongly expressed in these cells. And I'm sorry, the second part of the question was
you see the neurons making synapses onto OPCs, or the OPCs kind of.
No, it's a great question. So the synapses are unidirectional. They're always neuron to OPC. There's no OPC to neuron synapse, but there's some evidence that OPCs do a lot of things that are myelin independent. A lot of that work has been led here by Joanne, who showed that OPCs can prune axons. A group at Cold Spring Harbor has shown that OPCs can prune synapses. And there is also the thought that, there can be regulation through paracrine signaling of neuronal excitability through multiple glial mechanisms, but also including by OPC. So I think our understanding of the role of the OPC in myelin independent ways is really just beginning. But the synapses are unidirectional, both in the case of the OPC and in the glioma. There's no
reciprocal synapses.
Yeah. And they're not spiking. The glioma cells don't spike. There's no action potentials, and the OPCs.
But the OPCs can, right?
There's some so maybe in the cerebellum, but not elsewhere, is kind of the general thought. There's one paper by Thora Karadattir and when she was with David Outwell that highlights some spiking function, and it's controversial. But yes, the cerebellar OPCs might be a little bit different.
I want to ask one last question.
Yeah.
And this is back to ground zero of the whole myelin plasticity thing and activating OPCs and so on to form. And that is, how much do we know about the lifetime of a quotes mature oligodendrocyte? Do they last forever, or?
Yeah. In the healthy nervous system, they're very long lived. So there have been, there was a pair in Nature Neuroscience, one story out of Yale, one out of Dwight's lab, just longitudinally watching oligos in the superficial cortex. They live, and they live for a very long time. And the caveat is that they can be killed, and they can be killed by astrocytes, especially. So certain states of neurotoxic astrocyte reactivity directly kill mature oligos, and that may be the mechanism in COVID and in chemotherapy related, loss of oligodendrocytes that is at play. But in the healthy nervous system, they stick around. But that doesn't mean that all their internodes are stable, and there's evidence for, decreased activity-dependent reduction in myelination. So there's a lot of plasticity at play that we don't fully understand. There's probably myelin pruning, and some evidence for that in zebrafish. So lots to learn.
So the old guys don't necessarily have to completely get out of the way for new guys to come along.
That's right. They've got arms out, they're making, 50 internodes, and they can change in an activity dependent way. Yeah. They can pull their arm back.
Okay. Well, let's see, unless somebody else has the microphone. Well, thank you very much.
Yeah.
Let's have another wonderful talk.